guys, welcome back. Today I'll do two different topics in this video. The first will be pyruvate dehydrogenase complex, which is the pre-step to the Krebs cycle. Now we'll start with where we ended in our last video with pyruvate, which is the end product of aerobic glycolysis, and it must be transported into the mitochondrion through the inner mitochondrial membrane before it can enter the citric acid cycle, because it takes place in the matrix. Now this is accomplished by a specific pyruvate transporter. Once it is in the matrix, the pyruvate is converted to acetyl coenzyme A by the pyruvate dehydrogenase complex. Now this complex is a multi-complex of three different enzymes. One is pyruvate dehydrogenase, also written as E1, and it can also be called decarboxylase. Then you have dihydrolipoyl transacetylase, which is E2. And dihydrolipoyl dehydrogenase, which is E3. Each one of these enzymes catalyzes a part of the overall reaction. Now, coenzymes involved. Now, the pyruvate dehydrogenase complex contains 5 coenzymes that act as carriers or oxidants for the intermediates of the reactions. Now, E1, it requires thiamine pyrophosphate, TPP. E2 requires lipoic acid and coenzyme A, and E3 requires FAD and NAD+. Deficiencies of thiamine can cause serious central nervous system problems. Now this is because the brain cells are unable to produce sufficient ATP through the Krebs cycle if the pyruvate dehydrogenase complex is inactive, right? And thiamine is a coenzyme, it's necessary for the complex to work. So Wernicke-Korsakoff, I hope I'm pronouncing it right, it is an encephalopathy psychosis syndrome, and it may be seen with alcohol abuse. The mechanism is such that in E1, the pyruvate is decarboxylated. So it loses a carbon dioxide molecule, and in place of the C of the carbon dioxide, you have the thiamine pyrophosphate attached. Now TPP gives a thiol group, which is an SH group. In E2, which is a transacetylase, it gives acetyl coenzyme A, which replaces the thiol group. Now going to E3, it is a dehydrogenase, so it contributes an NAD to the electron transfer chain, which gives us 3 molecules of ATP. And from here we have acetyl coenzyme A from pyruvate going into the citric acid cycle. Now we'll speak about the regulation. So in addition to the enzymes that participate in the conversion of pyruvate to acetyl coenzyme A, the complex, it also contains 2 tightly bound regulatory enzymes. So we have pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphatase. PDH kinase, it phosphorylates and thereby inhibits E1. So phosphorylation of this enzyme will inhibit E1, whereas PDH phosphatase dephosphorylates and activates E1. So we have allosteric modification and covalent modification. Now let's talk about inhibitors. So the kinase itself is allosterically activated by ATP, acetyl coenzyme A, and NADH. Therefore, in the presence of these high energy signals, the pyruvate dehydrogenase complex is turned off. Now activators, so calcium is a strong activator of the PDH phosphatase, stimulating the E1 activity. Pyruvate is a potent inhibitor of PDH kinase. Therefore, if pyruvate concentrations are high, E1 will be maximally active. Why? Because PDH kinase tends to inhibit E1, and if pyruvate is a potent inhibitor of kinase, then it activates E1. I hope this is clear. If you are not able to understand what I mean by covalent modification and allosteric effect, please do head on to my video about enzymes. And with this, we are done with PDH. Now we will move on to the Krebs cycle. It is also known as the citric acid cycle or the tricarboxylic acid cycle, and it is the final pathway where the oxidative metabolism of carbohydrates, amino acids, and fatty acids, they converge and they're converted to carbon dioxide. It occurs in the mitochondria, in the matrix, and it is an aerobic pathway. Okay, our mnemonic for the Krebs cycle is children in kindergarten. C with a 2 because we have two S's and they're very similar. Friends more often. So children, C for citrate, I in isocitrate, kindergarten for alpha ketoglutarate, C succinyl coenzyme A, 2 is for succinate, friends is fumarate, more is malate, and often is oxaloacetate. And then oxaloacetate combines with acetyl coenzyme A to start the cycle again. Okay, the first enzyme is citrate synthase, which tells you that citrate is being produced, and the reactants are oxaloacetate and acetyl coenzyme A. The second enzyme is a conitase because it's like an isomer with isocitrate and citrate. And the third enzyme is isocitrate dehydrogenase, and this converts isocitrate into alpha ketoglutarate. The next enzyme is alpha ketoglutarate dehydrogenase complex, which converts alpha ketoglutarate into succinyl coenzyme A. And the next enzyme is succinate thiokinase, which converts succinyl coenzyme A into succinate. The next one is succinate dehydrogenase, converting succinate into fumarate. Next one is fumarase, which converts fumarate into malate. And in my last video, I mentioned the change of malate into oxaloacetate is by the enzyme malate dehydrogenase, and then the cycle repeats. You need to know that we lose an ATP in the third step. Carbon dioxide is released in steps 3 and 4 by both of the dehydrogenases, which was isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase complex. GTP is received in step 5 between the succinates. FAD is in step 6 from, you can remember, F going into fumarate, and NADH is in steps 3, 4, and 8. Remember, NADH comes from dehydrogenases, so isocitrate and alpha ketoglutarate dehydrogenases, and step 8 is malate dehydrogenase. So remember, the 3 dehydrogenases is for NADH. Now moving on to the energy yield. So we have 3 NADH from the cycle, and we know that 1 gives us 3 ATPs, so 3 will give us 9 ATPs. We also have 1 FADH2, so 1 will give us 2 ATPs, right? So that's 11 ATPs. We get 1 GTP, which is equal to 1 ATP, so 11 plus 1, 12 ATPs. And we know that we have 1 NADH from our pyruvate dehydrogenase complex in E3, and that 1 NADH will give us 3 ATPs, so 12 and 3 will give us a total of 15 ATPs produced from the complete mitochondrial oxidation of 1 molecule of pyruvate to carbon dioxide. And 2 molecules will then give us 30 ATPs because 2 molecules of pyruvate are received from glycolysis. And we revealed that aerobic glycolysis in my last video releases 8 molecules of ATP. So in total, we have 38 molecules of ATP from 1 glucose molecule. Okay, the last part of this video is about regulation. So in contrast to glycolysis, which is regulated primarily by phosphofructokinase, the The citric acid cycle is controlled by the regulation of several enzyme activities. Now, the most important are citrate synthase, which is subject to product inhibition. What does this mean? That the product of this enzyme, which is citrate, inhibits its activity. And isocitrate dehydrogenase is inhibited by ATP and naDH, and it is activated by ADP and calcium. And the last one, alpha ketoglutarate dehydrogenase complex, it is activated by calcium and it is inhibited by naDH and the product, which is succinate coenzyme A. Okay, that is it for this video. I hope it was clear. I'll see you in the next one. Take care. Bye.